Hi guys, welcome to another lecture in the Anatomy Physiology lecture series. This one is muscle physiology. It's a paired subject that goes alongside nervous system, which we've just completed, and makes up the second section of your AMP test or exam on nervous system and muscle phys. As you go through this, you'll want to keep this document here handy, which is the mock questions document, the same document that your nervous system information was recorded on. You'll see below the nervous system stuff is section 2 muscles, and we'll be filling in this stuff as we go, as we come across the pertinent points. And of course, this information becomes your study sheet, leading towards the exam on nervous system and muscle phys. So we're going to be covering a bunch of stuff as we go through this lecture here. These are the key lesson objectives. We're going to look at what muscle does for us, how muscle fires, so the link between the nervous system and the muscle. We'll then look at the microstructures involved with muscles. So we'll start from the outside and then work our way in right into that sort of micro structure invisible to the naked eye type contractile elements of muscle. And then we'll learn the sequence of muscle contraction called the sliding filament theory. We'll finish up with some discussion around fiber types, fiber allocation. Of course, we're talking about fast twitch, slow twitch, and intermediate fibers there. And then we'll top off the lecture with some factors that would affect your muscle contraction strength. So we'll start with functions. They can be broadly categorized into 4 key components there. The first one is force generation for movement. So that could be walking and running and moving your body from A to B, or it could simply be manipulating your environment, and even things like breathing, which are important to keep us alive. These are all muscular movements that occur within the body, either unconsciously or consciously, and are vital to our survival, and also make our time on Earth a bit more enjoyable as we can move and manipulate our environment to some degree. Next is posture. We have muscles that are primarily there to fight gravity and stop us folding to the ground, so they keep our head up. They support our legs when we're standing and make sure that our upper torso remains upright. So yeah, these guys are constantly working even if we're not. Heat production. Most of the body's warmth and temperature comes from muscle, either directly through voluntary muscle contractions that produce enhanced metabolism and therefore a byproduct of that is heat, or simply just the metabolism that keeps our muscles alive. Our muscles are the key metabolic generators in the body, and therefore as a waste product of their metabolism we keep a constant sort of heat within the body. So we know that our core body temperature is around 37 degrees Celsius, and much of that is maintained by muscle tissue. And then protection. Any entry or exit to the human body has a protective element with muscle, usually a sphincter type muscle that is able to close shut. So we're talking eyes, we're talking throat, we're talking even ears to a degree, and some of the other orifices you're probably aware of. And not only that, muscle helps protect areas where there aren't bony protection. And most mammals have a soft underbelly with a lot of guts underneath, and we have 4 layers of abdominals there to help protect that. They're not always in the greatest shape, so the body also will help protect by layering that area with fat as well if it doesn't believe the abdominals are strong enough to do the job. So we'll just fill out the first of our questions on the mock question sheet here. What are the 4 major functions of skeletal muscle? The first is movement. The second is posture. The third is heat production. And the fourth is protection. So one done there. Pop back into the Prezi here and continue. So what we're looking at here in this picture is a muscle. You can see an entire muscle that's been sort of harvested here. We've cut through it cross-sectionally there. And what you can see is the fibers inside this muscle are packaged into what we call fascicles. And these fascicles are made up of different types of fibers. So we could have a slow twitch fiber, bundle here, a fast twitch fiber bundle here, intermediate bundle here, and that would depend on your genetics and where the muscle is and what its function is. So we pull out one fascicle, and then we see the individual fibers. Just to give you an idea of their size, we're talking here about an individual fiber that's somewhere around the width of a human hair. So you can imagine that this picture that I'm showing you here is very much simplified in the sense that you could almost count these fibers here, but we would be talking bundles of hundreds or thousands of fibers within these fascicles here. What we want to look at here is the connection point between what we've just studied, the nervous system and the muscle fibers. And in this case, you can see a motor neuron coming in, and you can see it branching out into its synaptic nodes there and plugging into each of the muscle fibers here. So that's going to bring the electrical impulses that cause the muscle contraction to occur, and that's the sequence of events we're going to start looking at next. So first and foremost, let's pull this back so we can see it a wee bit. We know that the meeting point between two neurons is known as a synapse, and a synapse included a presynaptic terminal, a synaptic cleft, a little gap, and the postsynaptic membrane. Okay, so we knew the process there was that an action potential or electrical impulse made its way down the neuron, reached the presynaptic terminal, and encouraged the release of neurotransmitters across the synaptic cleft. And that caused some channels to open on the postsynaptic membrane where positively charged sodium was able to make its way in. Now the same exact process occurs when we have a neuron meeting a muscle. The only difference in this context is that the postsynaptic membrane, instead of being the dendrite of another neuron, is now a muscle. And in fact is a membrane that is wrapped around the muscle fiber, which I'll show you in a second. Instead of being known as a synapse, this meeting point between the muscle and the neuron, if we just go back and look at this picture here, this meeting point right here is known as a neuromuscular junction. So the nerve meets the muscle. There's still a synaptic cleft, there's still the acetylcholine or the neurotransmitters in there, and of course the pre and postsynaptic membranes still exist. They're just different things. One is a nerve and one is a muscle. So we would never talk about individual muscle fibers firing because that doesn't happen in the human body. So what we talk about instead is a motor unit, and that is we follow one motor neuron and we look at all of the muscle fibers that it might contract. Okay, so in the case of some of the bigger lower leg muscles or lower limb muscles, we might have a 1 to 500 ratio there, being that one motor neuron can fire 500 muscle fibers. In the calf muscles, that might be closer to 2,000 fibers. So one motor neuron traveling down there fires 2,000 muscle fibers. Okay, so a motor unit describes a pocket of fibers that fires when a motor neuron sends enough electricity down there. So it consists of the motor neuron, the muscle fibers that it attaches to, and of course the meeting points between the motor neuron and the muscle fibers known as the neuromuscular junctions. And here's an example of a motor nerve unit. We've got the one motor neuron coming down, and you can see it's plugging into multiple fibers, but these fibers are all red, meaning that only one type of fiber can be fired by this one motor neuron. In this case, it's very much simplified again. There are 3 fibers being activated, and they're all red, and those could all be, for example, fast twitch muscle fibers. It would take another motor neuron to come in and plug into the grey fibers to get them to fire. Okay. Of course, the more motor neurons that send messages to fibers and the more fibers that fire, the stronger the contraction is going to be. So the process leading to muscle innervation, just like what happened with the neurons, we have a motor impulse, same as an action potential, coming down the motor neuron, arriving at the presynaptic terminal of the neuromuscular junction and causing the release of neurotransmitters across the synaptic cleft. The neurotransmitter in this case is called acetylcholine. It's not important that you remember that. That just happens to be the specific one involved in this nerve-to-muscle communication. The acetylcholine goes across the synaptic cleft, binds to receptors on the muscle fiber surface, opens up some channels, and a new charge is then transmitted through the muscle fiber, hopefully enough to cause it to contract. So just because electricity arrives at the muscle fiber doesn't mean it's definitely going to contract. It's still really dependent on whether the amount of electricity that flows in through the sodium entering the channels, as to whether or not it reaches the threshold of the muscle fibre. And if the threshold is reached, just like the nervous system situation, the all or nothing principle states that if the threshold is reached, in this case the muscle fibre will contract 100%. So just like the nervous system, if the threshold is reached, muscle fibres can only contract 100%. There is no half contractions that occur in a fibre. There is the neuromuscular junction. We can see the endpoint here of the nerve, the motor neuron. This is the synaptic knob or presynaptic terminal. We have a little synaptic cleft going on here, and then we have an outside sort of skin layer around the muscle fibre. So here's the muscle fibre contractile elements here, and it has this kind of skin membrane over the top called the sarcolemma, which of course will conduct a new electrical impulse along the muscle fibre, causing them to contract. So we're going to learn a little now about the layers of muscle. We'll start from the outside and move away in, but muscles are essentially like onions. They have layers that we can strip away, and we'll get to the very innermost layers, and that's where the contraction of muscle occurs. And if we can understand how one tiny little component within the muscle contracts, we can then apply it to the entire muscle and understand how a full muscle contraction would occur. So we start with the outside. If you were to pull a muscle from a human body by snipping the tendons off at each end and just lump the muscle out, you'd find that it has a protective outside layer on it called fascia. Fascia is a protective tissue that surrounds all muscles, bones, nerves, blood vessels and other structures. Its main role is to protect and of course separate these different structures so that they don't grow into one another. So obviously at 37 degrees inside, it's nice and warm and conducive to growth. And of course this is all living tissue, so if it was to stick together or touch together, it would effectively graft. So it provides the structural integrity, it keeps things separated and able to work in their own units so that we don't graft them together. So there's the muscle, here's the bone, and here's the tendon attachment, and then this sort of clear membrane that's over the top. So I call it the human glad wrap that's sort of holding everything in place the way it should. We then have tendons, which of course the connection between the muscle and the bone, and here we see the largest tendon in the human body, or the thickest tendon, which is the Achilles or calcaneal tendon, attaching to the heel bone or calcaneus. And you can see that the muscle fibers simply graft or run into these collagen fibers, and these are very strong and very difficult to break, which serves their purpose really well. Without this link, of course, the muscle would not be able to pull on the bone and the skeleton wouldn't move. So tendons connect muscle to bone. I'm just going to pop into our mock questions just to answer these couple of questions here to describe fascia's structure and function. Fascia is a protective, connective tissue that surrounds muscles, organs, bones, etc. So it's a protective, connective tissue surrounding muscles, organs, bones, etc. And then number 3, what attaches muscle to bone? The answer is tendons. So let's have a look at muscle itself now. So again, a similar picture to what I just showed before. We have a full muscle here. We've cut through it cross-sectionally. We now look at the fascicles of different fibers, so bundles of fibers in here. We pull out one fascicle. We can see all of the different fibers in here. Okay, and then we pull out one fiber. So at this point, we have our single muscle fiber. Okay, we see it here. And this, like I said, is practically invisible to the naked eye in some cases, about the width of a human hair on a bench top. Now you can see that that's not the smallest part of the muscle. We see there are even more cylindrical rod-like fibers inside this. We pull one of them out. This is known as a myofibril. And then even more beyond that. And these are the tiny little contractile elements of muscle that we've got to get to. So from that we can gather that muscles are made up of a large number of individual structures called fibers. Inside those fibers are even smaller structures that we'll get to know. Each muscle fiber ranges in diameter from about 10 to 80 micrometers, which is millionths of a meter, so they are practically invisible to the naked eye in isolation if you just had one. However, they're as long as the individual muscle is in some cases. So some of those long quadricep muscles or hamstrings could be over 35 centimeters long, an individual muscle fiber. The number of fibers present in a muscle will depend be dependent on its size and function and the job that it's designed to do. So, I mentioned a skin-like substance that wrapped around an individual muscle fibre, and here we have an individual muscle fibre here, and you can see this kind of skin-like membrane over the top. And this is known as the sarcolemma, and it's this sarcolemma that effectively is the postsynaptic terminal in the neuromuscular situation. So the motor neuron comes down and plugs into this surface around the muscle fibre. Inside one muscle fibre we see even more tiny little fibres here, and these were called myofibrils. And as mentioned before, an even smaller array of fibres called myofilaments, which are the contractile properties of muscle. In between these myofibrils that we see here is these sort of yellow gelatin-like substances. So we'll dive in for a closer look, this sort of yellow stuff here. And that's actually called your sarcoplasm, and that's where your fuels and oxygen are stored. So this is where your muscle glycogen and your amino acids and your muscle triglycerides are stored for energy. So we've got complete access to them here to create ATP and fuel. Okay, the biggest word we're going to learn in this process is the sarcoplasmic reticulum. And that's this network of tubes that you see here, these little tubes underneath the sarcolemma here, all these little tubes here. And I want you to think of this as like a power grid. So if the motor neuron is the power cord that comes from, say, the Great Lakes, the Benmore Dam and that kind of thing, it's bringing in power to the city. The myofibrils inside the fibre are the city, and of course we know that we pass this electricity around the city via power lines and cables, and these would be the power lines and cables. When the new charge is stimulated on the postsynaptic membrane, it sends these electrical signals right down the myofibrils here through this network of tubes, and this is known as the sarcoplasmic reticulum. Now, its key role that you need to be able to link together here is that the sarcoplasmic reticulum is a storage site for calcium, which is an essential component for muscle contraction, which we'll get to in a moment. So right now, all we need to think of is when the motor neuron stimulates the muscle fibre, it generates a new electrical charge through this network of tubes called the sarcoplasmic reticulum, and that causes calcium to be released deeper into the muscle fibre. Now you can also see on the sarcolemma, this is a good picture to see these little holes. This is where the motor neuron would essentially come and plug in its synaptic knobs. So that's where the connection between nerve and muscle, or the neuromuscular junction, occur. It plugs in to the sarcolemma, stimulating some electricity to run through this network of tubes and releasing the calcium. So we've established now that each muscle is made up of hundreds to thousands of individual fibres packaged into fascicle bundles. Each fibre has its own little fascicles inside it of myofibrils, these little cylindrical fibres here. And then we're learning that even these have more and smaller little myofilaments inside them. So let's have a look. This is one almost invisible to the naked eye skeletal muscle fibre. And this is one myofibril that we're pulling out of it. And if we can understand how this myofibril contracts, then we can apply that to the entire muscle, and we can understand things a lot clearer about how muscle contraction occurs. So we're going to focus on one segment of this myofibril, and as you can see, it's broken into repeating subunits all the way along its length, and it gives it this kind of striped visual look, if you like, and that's to do with the overlapping of the two minute myofilaments in here, one called myosin, which we're going to know as fatty, and then one called actin, these little blue lines here, which we'll know as skinny. So we've got fatty and skinny. And you can see where fatty and skinny are overlapping here, it's a bit darker in the muscle, and where they're not overlapping, it's a bit lighter. And this is what gives striated or skeletal muscle its stripe-like appearance under microscopic conditions. So the fibre is the largest of the microstructures, then the myofibril, then we break that into subunits called sarcomeres, and in a sarcomere we find fatty and skinny, who are the guys that are going to interact and cause muscle contraction. So we'll just pause there for a second, and I want to go back into our mock questions and just answer this question here. Rearrange these structures in order from smallest to largest. Well, the smallest is actin, or skinny, the myofilament. Then myosin, or fatty, the second myofilament. Then we've got the myofibril, which is the third biggest structure in this setup. And the largest structure in this setup is the muscle fibre. And then we see it there, largest, second largest, third largest, and the smallest. So let's learn a little about how these guys interact. So fatty and skinny, actin and myosin, these are the guys that interact with each other and cause muscle contraction to occur. As we mentioned, actin is the thin little protein molecule here, and myosin is the fat one with these little arms that you can see grabbing on. So this is a better sort of picture of what's going on. It's a bit of a 3D model rather than a 2D model. You can see that these myosin heads, that we call them, these little grabby things here, are actually all around the myosin fiber. Now the actin up close is actually made of little protein molecules that we see here, these little red blobs, and there's a few other things involved in this too that we'll look at soon. But basically what wants to happen in a muscle contraction, these little heads want to jump up and grab onto actin and pull actin from each end towards the middle. And that's going to cause a minute contraction of the sarcomere. So if we go back to the picture here, if this happens, and we see down here, if these little arms pull towards the middle and these little arms pull towards the middle, that's going to shorten the sarcomere fractionally. So each of these little sarcomeres along the length of the myofibril, there are thousands of them, are going to shorten a minute amount, and that's going to pull the muscle from each end towards the middle, making the muscle bulk out in the middle. And of course, if the muscle is attached to a bone at the other end, it's going to pull that part of the joint closer to the anchor point, or the origin. So when these guys make connections, these little guys make connections with the actin, if we look at this guy here, we can see that he's making a connection. This is known as a cross bridge. But actually in its resting state, before the electricity has arrived to fire muscle contraction, these guys are unable to grab onto actin. They're prevented in doing so by this barrier that you see here, this white tube that's running through here. And this is covering the bits of the muscle, of the actin that the myosin head wants to grab onto. So we need something to open those doors. And that something is that important mineral, calcium. We'll just discuss the events that occur there just now. So here's actin, and actin has a couple of key players on it. Here's the barrier, which we see here, and this barrier is known as tropomyosin. So this is a gate that is closed until stimulation from the nervous system occurs. And on tropomyosin, we have this little guy here who's a bouncer, and his name is troponin, and his job is to facilitate the opening of this barrier if he's paid off in the form of calcium. So what we need to happen is we need calcium to be freed from the sarcoplasmic reticulum to come down and bind to the bouncer troponin, who will lift off the tropomyosin barriers, and that will allow myosin access to actin, and then the cross bridges can form that you're seeing here, and these little guys are then going to pull actin towards the center of the sarcomere, causing muscle contraction to occur. So we're going to look at how that muscle contraction occurs now. So we've been talking about the actin and myosin and how they interact, and it's time to have a look at that action in a sort of physical sense. So I am actually going to play this little video here and just talk over the top of it about the sequence of events that occur during the slide and filament theory. So yeah, let's have a look at this here video, and this should hopefully make things a lot clearer. So there's a little bit of preamble here while the video loads up. And what we're going to see is a little dude working on his guns before the video dives in. And here he is. So he's going to start a bicep curl, and we're going to dive into the muscle. And we're going to see that this muscle is made up of cylindrical structures called muscle fibers, and there they are, and they're bound within their fascicles here. So we see bundles of muscle fibers here, and the striped nature of them, of course, indicating where actin and myosin are overlapping in the dark sections, and in the light sections where the actin and myosin is not overlapping. So we're going to pull out one fascicle here. So as we pull along, there's one fascicle. Okay, and from there we'll pull out one fiber, and there is the single muscle fiber. And we can see inside that there are multiple smaller fibers, which we now know are myofibrils. And those myofibrils are broken up along their length by these red lines known as Z lines, and between the two red lines we call this one sarcomere. So as we pull across, we'll isolate a sarcomere, and the video will start showing now how the sarcomere contracts, and if all of the sarcomere along the length of the myofibril contract, that's what leads to muscle fiber contraction. So we'll dive in and have a closer look. Here we can see myosin in red and actin in blue, the thin filament and the thick filament. And as we dive in, you'll have to excuse the jerky nature of the video here, just the old internet playing up a little. You can see that the myosin of the myofibril here has the little myosin heads, these are called here, and these guys are interacting with the actin by jumping up and pulling the actin. Now, the way it's set up at the moment, you can see it's just going back and forward. That's not really conducive to what happens in the real sense, but it just gives you an indication of the sort of movement of the myosin heads at this point. Okay, up above you can see actin in blue, and you can see this tube-shaped tropomyosin barrier that exists. Now you see ATP ready and waiting on the myosin head. Can't do anything yet because the barriers are closed, but here comes calcium. Watch these barriers up here now that calcium has arrived. Tropomyosin will now roll away, exposing the active binding sites. Now watch the ATP on the head here. This is energy being combusted, and bang, off goes the myosin head and makes a cross bridge with the actin. We need more ATP to rotate it into position. Up it goes again and pulls again. And a myosin head will do this about 5 times every second, depending on the fast or slow twitch nature of the muscle fiber. By doing this, and these guys working independently together, all of these hundreds of little heads, they're pulling the actin towards the middle of the sarcomere. And if that happens from both ends, then of course the muscle will shorten. Now we're seeing tropomyosin has effectively dropped back into its covering position on the actin, and that means the myosin head can't grab on anymore. So if that happened and all myosin heads dropped off, we'd see all myosin heads let go at once, and of course the actin would just return back to its normal state, and the muscle would relax pretty quickly in that sense. And the poor old fella here is going to relax his muscle very quickly, drops the old weight, and the result is not great for him. So a couple of key points just coming out of that. We'll just skip on to the next one there. And the key points are this. First and foremost, we've got to understand the mechanism of action of the myosin heads. And what's happening here is these myosin heads are working somewhat independently. If you consider a situation where a group of people are in a single line and they're passing a rope over their head, then what needs to happen is some people need to have their hands on the rope at all times. If they all let go at the same time, then the rope obviously drops down. So you can see here a couple of myosin heads are attached and a few more are rotating back for their go next. So we're always going to have this constant sort of attachment happening. A reminder that the meeting point between the myosin head and the actin is known as a cross bridge, and the pulling action that we see occurring where we pull the actin towards the middle is known as a power stroke. Again, in its resting state, the myosin can't get access to actin because the parts that it wants to bind to are covered by the tropomyosin molecule, the tube-shaped barrier here. And we didn't see it clearly in the video, but we saw calcium arrive and we saw the tropomyosin lift off, but actually the calcium would have bound to troponin, which would have lifted off the tropomyosin, allowing the activity to happen. And we saw in the video that all of these myosin head miniature contractions that occur, these cross bridges and power strokes, are fueled by ATP, which is of course derived from the fuel that we eat. So from that we deduce that the two key substances, of course, for muscle contraction are calcium and ATP. This is probably a good time to whip back into our mock questions here, and we can answer a few questions about this whole process now. So what have we learned so far? What is the sarcoplasmic reticulum's role in muscle contraction? And the answer to that is it holds calcium and releases calcium. Okay, so we'll write in there, it holds calcium and releases calcium when stimulated by a motor neuron. And then we can go deeper in and look at what role does calcium play in the muscle contraction, number 7 here. We know that it binds to troponin on actin, and this lifts tropomyosin, allowing a cross bridge to form. I'll repeat that again. So calcium binds to troponin, which lifts tropomyosin, allowing a cross bridge to form. And this whole process I've just described to you, where actin and myosin slide past each other, this is the theory of muscle contraction, and this theory of muscle contraction is called the sliding filament theory. So we can add in here for number 8, what does the sliding filament theory describe? It describes muscle contraction, is the answer to number 8. What are the two parts that make up a cross bridge? The answer to that is the myosin heads and actin. And then number 10, when a muscle shortens, it is achieved through repeated cross bridges and power strokes. Okay, so that's where the myosin and actin are interacting. And we can answer this last one here, number 11, what two things are needed to continue muscle contraction? What two substances that could read? And they are, of course, calcium, which lifts the barriers, and ATP, which fuels the whole movement. Alright, so a lot of good learnings out of the year. You can see the types of questions you might be asked come exam time. We'll open this back up again, and we'll see a bit of revision of this process as we go through, and even another video coming up soon. So there it is, the role of calcium. It binds to troponin, which is the bouncer, and can be found on actin, and troponin lifts up tropomyosin, the barrier, and that exposes the parts on actin that myosin heads would like to form cross bridges with, and it starts that contraction process. When the cross bridges form and the power strokes occur, the two filaments slide past one another, and actin is pulled towards the center of the sarcomere from both ends, and that of course shortens the muscle. That tilting of the myosin head that pulls the actin filament towards the middle of the sarcomere is called a power stroke, as we have determined. Okay, so that is effectively the sliding filament theory. A little bit of revision on that now because it is a really important point, and that will finish the first of our muscle physiology lectures, and we'll continue with the next bit after. So nice short one just to introduce the concept, and we'll do a few practice tasks on this in the next video too. Alright, the importance of ATP. Remember that ATP is vitally important for this process because that provides the energy for the power strokes to occur. The two key substances, as we've determined, are calcium and ATP for this process. So here we see the ATP binding site, which is right on the myosin head, and causes that activity that starts the muscle contraction. Now immediately after one power stroke, the myosin head of course breaks away, rotates back into its starting position and grabs the actin again further along, and in this way it slides the actin towards the center. Repeated attachments and power strokes and those reattachments fueled by ATP will cause that filament to slide past, actin filament to slide past myosin. Of course that's the sliding filament theory and how the muscle contracts, and we understand that is happening in one sarcomere, but it's happening in all the sarcomeres along the myofibrils and all the myofibrils within a muscle fibre, because we know that not just one fibre contracts together. So hundreds and hundreds of thousands of these little myosin heads grabbing on and working away during a contraction, which we take for granted. So it's a pretty special process. Another little video here that just cements that knowledge. Okay, so we will again have a look at this one because I think there's some good learning in it, and I'd like to guide you through it again. So this will recap the process. And here we see the myofibril myosin portion, and here we see the actin myofibril down here. And we can see here the ATP molecule arriving. And that's going to start the process of cocking the myosin head back. And then of course we need the arrival of calcium Here we see troponin. Okay, and we see tropomyosin here. So right now it's just showing that active binding. Now here comes the ATP again, and we see the cross bridge forming and the power stroke occurring. Now remember, none of that can happen without the arrival of calcium. So here's calcium in red. Calcium's going to bind to troponin, so it rotates those tropomyosin out of the way, and that allows the cross bridges in action to form. So hopefully that gives you a better understanding of the process required for that to happen. Now here, just to finish off, you can see this is the starting length of the muscle with actin and myosin overlapping here, but good space available for contraction to occur. So what we do is we watch the Z lines here, the black Z lines, and you can see as the process continues and the myosin pulls the actin strands towards the middle, they get shorter and shorter, the Z lines come closer and closer together, and if that occurs across the whole length of the muscle, we're going to see a meaningful muscle contraction occur. So the very last slides we'll look at is what happens when it's all done and you want to stop the muscle contraction. We've already seen in the video, the first video, that if you let go all at once, then a weight that you're lifting or the movement that you're doing will simply cease, and the muscle will drop very quickly back into its resting position. That's not usually what occurs in the gym-based sense. We know we don't usually just drop our weights at the end of the set. Some people do. But really we're looking to control that eccentric portion of the lift as well. So what happens there? Calcium and ATP, of course, drive the muscle contraction process. After the muscle contraction, and the message is received from the motor neuron to stop the contraction, any calcium that hasn't been used is pumped back to the sarcoplasmic reticulum, and when the calcium disappears, of course, then the barriers drop down again, and we just use one last molecule of ATP in each of these myosin heads to break away one last time, and the muscle fiber will relax. So during an eccentric contraction, we want to control the down phase of the muscle contraction. What happens there is certain muscle fibers will be told to stop working, and these processes will occur while others continue to work, and that way gravity starts to win the battle, but in a controlled fashion the weight would be returned back to its starting position. You can see here in the last slide that we're going to deal with today, acetylcholine was the neurotransmitter that crossed the synaptic cleft that got the message across and caused the muscle fiber to fire. Now we release an enzyme called acetylcholinesterase, which breaks down any remaining neurotransmitters that are crossing the gap, and therefore the message no longer gets through. As mentioned, the calcium goes back to the sarcoplasmic reticulum, ATP breaks the links between actin and myosin, and troponin and tropomyosin return to their resting state, and of course the muscle relaxes. So that's plenty for this installment right now. There's some questions on the little quiz that I've put on Moodle for you to work your way through to answer some of these. You won't be able to answer them all yet because we've got another half of this lecture to go. The next time we talk, we'll be discussing muscle fiber types and your genetics and how that's led to the types of muscles that you have. Thanks for listening in.